This section installs the single frame the whole chapter runs on. Read every technique as one coordinate on a single axis. How defined the manipulation of living matter is. At one pole sits Teosinte, bred into maize over 9,000 years. That work needs no biological knowledge and moves blind. At the other pole sits a cell whose genome is chemically synthesized from cataloged parts. That work needs deep knowledge, but it buys total predictive control. Synthetic biology is the directed collapse of biological engineering toward the defined pole. It imports the engineer's grammar, hierarchical design, modular reusable parts, and standard interfaces onto a substrate that nature already stocked with sophisticated parts. The chapter's job is to install this axis as the frame through which assembly, circuits, and the synthetic cell all become the same move at different resolutions. This section states the single claim that turns biology into an engineering discipline. Life becomes an engineering substrate the moment you stop treating its parts as given and standard interfaces. That is the chapter's whole claim. Synthetic biology is engineering, a discipline of design rather than a science of observation, and it imports the habits of engineering, hierarchical design, modular reusable parts, isolation of unrelated functions, and standard interfaces. Its single axis of progress runs from undefined to defined. At one pole sits Teosinte, bred blind over 9 millennia. At the other sits a genome assembled wholly from synthesized DNA. The peculiarity, unlike architecture or software, is that nature already stocks the parts bin, so the work is mining found components, promoters, haptomers, terminators, and recomposing them under predictive control. Everything downstream, DNA assembly, circuits, the synthetic cell, is this one shift made concrete. This section explains how the first living cells could have emerged and why life splits into separable parts. Life factors into 3 separable parts: a machine that reproduces in cuts an inside from an outside, a program that replicates, and a metabolism that couples the two. This is the same chassis and cookbook split that synthetic biology later makes real when a transplanted genome reboots its host cell. The move that makes the argument go through is Dyson's. Reproduction, a metabolism making similar copies, must precede replication, templates making identical copies. So at least 2 chemically independent origins were needed, not one common ancestor. Read backward. As a retrodiction from the biochemistry still living today, the first cells resolve into a lineage of compartments, mineral surfaces first, then RNA, then lipid-bordered phagocytes. Information crept into these physico-chemical assemblies incrementally. It was never poured in whole. This section explains why life splits into 2 bodies. Life factors, and the factorization is the chapter itself. Danchin's primitive is to separate the machine from the program, a chassis that reproduces, making a similar copy. An approximate one is distinct from a cookbook that replicates, making an identical copy an exact one. The 2 are coupled by metabolism, which steers matter. The surprise that does the real work is Dyson's ordering. Reproduction must predate replication, so life demands at least 2 chemically independent origins, not 1. This is doxa rather than aletheia, opinion rather than settled truth, but it is structurally forced. Compartmentalization is the thread stitching them together. First a mineral surface, then RNA, then a lipid membrane. Each is a container that selects what reacts. From this you can predict the rest. Any scenario asking for a single event or for an exact copier first is already wrong. This part introduces the surprise the whole chapter runs on. The same genome, different cells, Every cell of a multicellular body carries identical DNA. The heterogeneity comes from which stretches are read, when and how much. The code is the same everywhere. Regulation is that decision layer sitting over a fixed program. Its cheapest lever is the entrance, transcription initiation, gating a gene before you pay to transcribe it. The operon's inducer and repressor logic is the anchor. Beta-galactosidase switches on only for lactose and only when glucose is absent. Every downstream stage is a further gate. Splicing, messenger RNA transport, translation, epigenetic memory. Each one is another point where the read can be blocked or let through. For a synthetic biology book, this is the point. These gates are precisely the handles an engineer grabs to command a cell. This section explains what makes cell types differ when every cell carries the same genome. Every cell in you carries the identical genome, yet a neuron and a liver cell share almost nothing in behavior. What makes them differ lies not in the sequence but in which genes are read, when, where, and how much. That is the whole chapter. Expression regulation is a control layer riding on an invariant program. Each mechanism it names is one gate placed somewhere along the single path from gene to product. The operon switch, attenuation, chromatin packing, the splicing that recombines exons, microRNA silencing, epigenetic memory, every one of them is A combines exons, microRNA silencing, epigene on that one path. Learn the path, and every mechanism becomes predictable. A gate can only sit at one of four places: DNA accessibility, transcription, RNA processing, or translation. There is nothing else to regulate. This section explains what the interactome is and the single conceptual move the chapter makes with it. Take a cell's every molecular contact, all of it together, and that is the interactome. The whole move of the chapter is to stop treating it as chemistry and start treating it as a graph. Molecules become nodes. Interactions become edges. The edge is directed when one party acts on another. A transcription factor acting on another. A transcription factor acting on another. That substitution is the entire payload. Once the cell is a graph, biology inherits graph theory, wholesale, degree distributions, modules, motifs, network comparison, none of which ever knew anything about proteins. Everything downstream is bookkeeping in service of that one topological reframe. Yeast. 2. Hybrid and tandem affinity purification with mass spectrometry. Measure the edges. Evolutionary and statistical methods predict them. All of it serves the single idea that the cell is what its wiring diagram does. This section explains why a protein is best understood by what it touches. It reframes the cell as a graph where the nodes are proteins and the edges are bindings, and it lets topology carry biology that sequence alone cannot. A node's degree predicts its essentiality. Its neighbors predict its function. A disease becomes a perturbed subgraph. The generative move is that you infer an edge without ever watching two proteins bind. Genes that fuse in one genome, that co-occur across genomes, or that co-vary in sequence, all betray a functional link. Evolution's bookkeeping substitutes for the assay. Relation precedes substance. Once the graph exists, its shape does the explaining. This is the same relational turn as the idea that an object is defined by what it does. The protein dissolves into its pattern of interactions. This section explains what the microbiome is and the sequencing inversion that made it visible. An organism you cannot grow in a dish. You could not, traditionally, sequence. Cultivation was the gate. Metagenomics removes the gate. You sample a habitat, You sequence everything at once, and you read the community's collective genome without ever isolating a single member. That inversion, from purify then read to read the whole, is what the word microbiome names. It means not the microbes alone, but their aggregate genomes and environmental interactions inside a niche, the human gut and the Sargasso Sea alike. The chapter's work is to show what follows from dropping one constraint. Drop the demand for a pure culture, and you expose the uncultivable majority that had stayed invisible. That same move turns gut ecology into a lever on personalized medicine. This section explains what metagenomics changes about the study of microbes. The whole move is to change the unit of observation from the isolate to the ensemble. Traditional genomics demands a pure culture, one organism grown clonally, its DNA read in isolation. The method is therefore blind to everything that refuses to grow in a dish, which is the overwhelming majority of microbes. Metagenomics deletes the culturing step. You sample a habitat directly, sequence every genome present at once, and treat the community in situ as the object rather than the individual cell. The anchor is the Sargasso Sea sample, surrendering 1.2 million new genes. Not one organism sequenced well, but a whole niche made legible because you stopped trying to separate it into parts. This section explains how a cell's behavior in time is written down and why one choice governs the whole chapter. A cell is its regulatory wiring in motion. Genes, proteins, and metabolites push each other's activity through time, and everything turns on a single choice: at what resolution you write that motion down. One system, but two languages for it. The first language is the Boolean formalism. It collapses each node to on or off and asks only what logic gate the network computes. This is coarse, but it scales to thousands of genes. The second language is differential equations. It tracks continuous concentrations changing at measured rates. This is exact, but it drowns once the unknown kinetic constants multiply. The pattern underneath is a resolution and extent trade-off. Detail and size trade against each other, and the size of your subsystem picks the formalism, not the biology. Everything downstream, the attractors, the oscillators, the switches, is this one dial, turned. This section explains how a cell's regulatory circuitry can be read as one dynamical system at two resolutions, and how the chapter gives you the rule for choosing which. A cell's regulatory circuitry is one dynamical system, read at two resolutions. The whole chapter is the rule for choosing which. Push toward large scale, and the sigmoidal input-to-output curve of every regulatory link collapses to a step. Genes are simply on or off. Boolean dynamics suffice. Push toward a few genes, and you keep the curve, paying in ordinary differential equations and rate constants you rarely know. So the choice of formalism is not taste, but a scale versus detail trade. Coarse graining is licensed precisely where the response is steep. That single move, one sigmoid, two limits, is what the chapter hands the rest of the book. This section explains eCell, the claim that a whole cell can be run as a simulation. Start from what a cell is. A cell is a massively parallel and massively interactive biochemical reactor, holding everything it needs to sustain itself. eCell is the claim that such an object is runnable. Every process, from enzymatic metabolism through transcription through membrane transport, is collapsed to a mathematical scheme coupled together and integrated forward in time. The move is reification of dynamics. A static pathway map becomes a program whose execution is the cell's behavior. The chapter's proof of concept is a self-sustaining virtual cell of 127 genes. It is Mycoplasma genitalium, pared down to the minimum, just stable enough for survival. What the chapter is for in the book's arc is this. It establishes the whole cell rather than one gene at a time as the tractable unit. Once you can simulate it, this section explains E-cell and why it reframes what it means to simulate a living cell. A living cell is a reaction network that runs itself. E-cell, short for electronic cell, treats simulation as integrating that network until it closes on itself, rather than copying the cell. Each process enters in its native mathematical idiom. Enzymatic metabolism enters as differential equations, gene expression as stochastic binding, and the whole cell is their composition. The claim that carries the chapter is that with modern compute, the hard part is conceptual. The bottleneck is choosing the right abstraction, not doing the arithmetic. The chapter's anchor is a self-sustaining virtual cell of 127 genes, the minimal set whose reactions mutually close, just stable enough for survival. Life, here, is a fixed point of a network rather than a parts list. This section explains SynBio-SS Designer as a compiler aimed at biology. You hand it a parts list, inducers, repressors, promoters, and coding regions pulled straight from the standardized BioBrick registry. It then mechanically expands that declarative specification into the full network of biochemical reactions, those parts in polarity. The separation. The chapter is for works like this. The practitioner states what the circuit is made of. The software derives how it behaves, automating the tedious, error-prone hand derivation of every binding, transcription, and degradation event. Non-intuitive network behavior becomes predictable without modeling expertise. The front end is a parts catalog. This section explains SynBio-SS Designer, a tool that turns a parts list into a full reaction network. Declare the parts and the reactions write themselves. Designer is a compiler. Feed it the molecular inventory of a synthetic construct, the promoters, the coding regions, the proteins and inducers acting on them, and it emits the full biochemical reaction network governing that construct's dynamics. The modeler, no longer hand, derives transcription, translation, multimerization, DNA binding, leakiness, and degradation. Those follow deterministically from the parts list, since a BioBrick's behavior is fixed by its type. The move that does the work is any compilers raise the specification above the mechanism, so the human states what is present and the tool derives what happens. There is a boundary. Designer generates the model, and executing it is a separate step. Simulating the network is its own task, whose cost scales with the size of that network. This section explains the founding move behind synthetic gene circuits. The whole idea is a living cell rewritten as engineered hardware. Take the abstraction hierarchy that electrical engineering already owns, where high-level behavior compiles down to concrete parts, and transpose it onto molecular regulation. Under that transposition, every circuit decomposes into 3 stages: a sensor, then a processor, then an actuator. The stages are built from parts chosen to be orthogonal, meaning no crosstalk, so that they compose cleanly. Once that holds, a request-like design, a circuit that finds and kills cancer cells, becomes a specification that descends layer by layer, down to descends layer by layer, down to DNA sequences. The 2 proofs of principle, the oscillator and the toggle switch, matter for a single reason. They are the first demonstrations that transcription behaves like components you can wire together. From there, you can predict the rest: logic gates, memory, analog computation, all of it, the same transposition carried further. This section explains why a living cell counts as a programmable substrate. A cell becomes programmable the moment you insist its regulation composes like electronics, an input sensor, a processor, an output actuator, wired together from parts that must not crosstalk. That non-crosstalk requirement is orthogonality. The chapter's whole move is transplanting the engineering abstraction ladder onto transcriptional control. You start at high-level behavior, recognize and kill cancer cells, and descend all the way down to the exact DNA sequence. Everything cataloged further on, the oscillators, the toggle switches, the Boolean gates, the memory, instantiates a single claim. Regulatory interactions are reusable primitives, and a cell computes when you compose them predictably. The list of devices is not the thesis. The thesis is that biology admits the same layered design discipline as a circuit. The metaphor is meant literally. It is the wire itself, not a simile about a wire. This section explains what a DNA origami nanorobot is at the most structural level. A robot is not metal. It is a loop. Sense the environment, process the reading, actuate. The chapter's move is to collapse that entire cybernetic triad onto a single folded strand of DNA. The nanorobot is a hexagonal clamshell latched by two aptamer arms. Each arm binds one target molecule, and only both targets present will unlatch the clamp. The structure itself is an AND gate, sensor and processor and trigger fused into one motif. Open, it spills its cargo onto the target cell. Here is the generative primitive. Robotics is substrate independent. Give any medium the sense, compute, act loop, and it robots. Anderson's combination lock is the whole idea. The winning combination is a molecular signature, and the opening is the therapy. This section explains why DNA is the natural substrate for a molecular robot. A robot is substrate neutral. It runs one loop, sensing, then processing, then actuation, wired together by cybernetic feedback. That loop is the same whether the body is steel or a folded strand of DNA. Bachelet's move is to notice that DNA is the one substrate where all three of those functions collapse into a single material. Watson-Crick pairing already computes. It also senses by way of aptamers, and it actuates through strand displacement and folding. So the machine need not be built first and then interface to biology. It is written directly in biology's own alphabet. This dissolves the drug control problem the chapter opens on. Arbitrary control over where and when a molecule acts demands a nanoscale computer that speaks molecules. Cells are that computer already, and origami lets you fold a synthetic one from the same code. This section explains why RNAi can serve as the whole control loop inside a living cell. Every engineered controller is the same. Three-stage composition, sense, then compute, then actuate. RNAi supplies all three inside a living cell, using nothing foreign to it. Endogenous microRNA and small interfering RNA concentrations are the input. Base pairing repression turns a molecular level into a logic variable, so the cell's own transcriptional fingerprint becomes readable state. Wire these repressions into Boolean gates, and the circuit reads which cell it is sitting in, then actuates conditionally. It is a cell type classifier written in concentrations rather than voltages. The chapter's thesis is that RNAi is the interface. It is the modular, scalable coupling between a synthetic circuit and the cell's native signaling, the coupling that makes computing over endogenous inputs possible at all. This section explains how one abstract pattern, the sense, compute, actuate decomposition of any information processing device, drops into a living cell. Sense, compute, actuate is the same three-block decomposition you find in any information processing device, and here it is placed inside a living cell. RNA interference is what makes that decomposition operative, and it does so for three reasons. Silencing strength is fixed by the base pairing complementarity between strands. A native microRNA's abundance becomes a readable logic input, and a transcript you design becomes the wire. Notice what this buys you. No sensor protein is added because cell state is already written in which microRNAs are present, and complementarity is programmable. Pattern matching, so the matching rule is something you specify. The chapter's thesis is that this turns RNA interference into a modular and scalable interface between engineered circuits and the cell's own endogenous signals. The cell reads its own molecular condition, evaluates a Boolean function on it, and actuates the result. The deeper point is the substrate. Independence of the sense, compute, act pattern, here shown in wetware. This section explains what a biosensor is at its root. A biosensor is one composition read twice. A recognition element that binds only its target, wired to a transducer that turns that binding into a macroscopic signal. Everything the chapter enumerates is a choice in two independent slots. Recognition, which is either catalytic or affinity-based, the affinity kinds being antibody, complementary DNA, or ligand. This is multiplied against transduction, which is electrochemical, optical, piezoelectric, or thermal. Hybrid names the seam itself. Molecular specificity carries no readout. Physics carries no selectivity, so the device is exactly their coupling. The one fragile step is immobilization, fixing the biological half to the transducer without killing its activity. The sensor is only ever as good as that join holds. Name a sensor's two factors, and you predict it. This section explains what a biosensor is at its most basic structural level. A biosensor is a composition of two independent functions. The first is a recognition element that binds the target selectively. The second is a transducer that converts that binding into a measurable signal. Everything the chapter enumerates, amperometric, potentiometric, optical, piezoelectric, thermal, is one choice on the transduction axis crossed with one choice of receptor. The whole taxonomy is just the product of the two. The word hybrid names the join itself. Its fragile hinge is immobilization, fixing the receptor to the transducer surface without killing its activity. Given the two axes, you can generate any device in the table yourself and predict which coupling step fails first. This section explains why the winning move in engineering, a long metabolic pathway, is spatial, using vitamin B12 as the worked case. A pathway of some 30 steps that leaks or poisons at every intermediate cannot be made efficient by improving any single enzyme. The winning move is spatial. Cobalamin, vitamin B12, the one vitamin-only prokaryotes build, is the chapter's worked case. Nature runs its most baroque biosynthesis microcompartments, but on locality. It hands each fragile, oxygen-sensitive intermediate straight from one active site to the next, a mechanism called substrate channeling. Then, downstream, it seals whole pathways in sight to the next, a mechanism called substrate channoecious microcompartments. Here is the primitive to carry forward. Throughput is set by where reactions happen, not only by how fast. Confine the volume and loss disappears. That same inversion is what makes these bacterial organelles reprogrammable into bespoke bioreactors. This section explains why vitamin B12 makes a metabolic pathway look like an architecture rather than a bare list of reactions. Nature's most convoluted biosynthesis is vitamin B12, the one vitamin that only bacteria and archaea can build. This chapter uses it to prove that a metabolic pathway is a piece of architecture. Reading it as a reaction list misses what actually governs the outcome. Here is the step that makes the argument go through. The intermediates never diffuse free. They are handed enzyme to enzyme by a mechanism called substrate channeling, and whole pathways are sealed inside proteinaceous microcompartments. Efficiency is therefore geometric, not merely catalytic. The variable you can engineer is spatial arrangement, not just the rate of each enzyme. This turns the naive metabolic engineering picture into something sharper. Control where the reactions happen. This is the chapter's place in the book's larger arc. Such a compartment is a reprogrammable shell. Swap the embedded pathway and the organelle becomes a bespoke cellular bioreactor. This section explains what the minimal gene set really is. What counts as minimal is set by subtraction. Whatever the environment supplies, the genome can drop. So the minimal gene set has no single fixed value. It is whatever remains after the environment pays for the rest. Minimal factors cleanly into two pieces. The first is a universal informational core. Replicate DNA, transcribe, and translate. This core is conserved across all three domains of life because copying the copier cannot be outsourced. The second is a variable metabolic shell, whose size collapses as the growth metabolic shell, whose size collapses as the growth medium grows richer. The step that does the work is this. Essentiality is relational. It is a property of gene times environment, never of the gene alone. This is why no universal minimal genome exists. There is only a conserved core wrapped in an environment contingent remainder. You can predict any case from this. Fix the nutrient medium, and the metabolic genes it renders redundant vanish. The translatome never does. This part explains why a cell's minimal gene set has no fixed size. Strip a cell down to the fewest genes that still keep it alive, and the lesson is that the minimum is a functional of the environment, a quantity set by the surroundings rather than a single fixed count. The chapter's move is to split the word essential into two pieces. First, an invariant informational core. Replicate, transcribe, translate, nearly identical across all life. Second, a metabolic remainder that shrinks as the niche supplies more for free. A parasite bathed in a host's metabolites needs almost no biosynthesis of its own. A free-living cell must build everything itself. So no universal minimal genome exists. There is only a fixed core plus a set indexed by what the world already hands you. Minimality is subtraction against a background, never an absolute count. This section explains why swapping the container beats tuning it when you need to hold enormous stretches of DNA. The move is to change what does the holding. E. coli keeps guest DNA as a separate self-replicating plasmid. That caps near 350 to 400 kilobase pairs, and it demands the fragment be circular before replication can begin at all. The BGM system exploits the natural competence of B. subtilis. Its membrane binds any double-stranded DNA. nonspecifically, threads it in as linear single strands and recombines it into the host's own 4,200 kilobase pair chromosome. The guest becomes part of the genome, so the capacity is the chromosomes, not a plasmid's. Whole mitochondrial genomes, giant engineered chromosomal segments, sequences that E. coli chokes on, all of these clone with little difficulty. When a substrate saturates, you do not tune it. You swap the container. This section explains a single structural move, making the host cell's own chromosome the vessel for cloned DNA, instead of carrying it on a self-replicating plasmid. Stop treating cloned DNA as a passenger riding on a self-replicating plasmid. Make the host's own chromosome the vessel. That is the entire move. E. coli imports DNA passively, so the insert has to arrive as a closed circle, and a plasmid buckles once it passes roughly 350 to 400 kilobase pairs. Bacillus subtilis is naturally competent. It actively drags linear DNA across its membrane and recombines it straight into its genome. Capacity therefore stops being the passenger's weight limit and becomes the chromosome's. A whole 3,500 kilobase pair cyanobacterial genome fits. Assembly turns into ordered stitching in situ, fragment after fragment, which is the domino method. The container is now a living cell rather than a ring of DNA.